I've just put up the agenda again so you can see what we're going to finish with. We've just got just over an hour to go till we finish at 12:30. So I'm just going to talk really briefly about how you can join a Waterwatch group. I've just put a link in the chat for all the groups in Mary Creek Management Committee area. So we've got 8 groups that are currently monitoring monthly. Most of them are just doing water quality testing. So all the water testing parameters we've just been through, they're doing them every month. Some groups are also doing water bugs regularly. And one of those groups is Trevor's group. So Trevor is the guru of water bug sampling. So if you do want to learn from the master, it is a good idea to go out with Trevor's group. So he's got 2 groups that are on that list. One's on the Mary Creek in Fitzroy North and the others on the Edgars Creek in Coburg North. So you'll see the groups cover a few different areas, but not everywhere. So someone asked about the Moonee Ponds Creek. We have got one group that monitors 5 Mile Creek, which is a small tributary of the Moonee Ponds Creek in Essendon North. But at this stage, that's the only group we have. So we only have funding to do monitoring for about 8 groups. So funding is required for each group, so I can go out as the Waterwatch coordinator and check each year with each group to see how they're going, that their site is safe, for training, and to for their water testing kits to make sure they have the right equipment, and to give them support in entering the data on the database and all that kind of thing. So at this point, we can't add any more groups. So the main thing to do is join an existing group. That's also the same thing for the Melbourne region. So the rest of the area, so I look after groups on the Darebin Creek, 5 Mile Creek, which is part of the Moonee Ponds Creek, Mary Creek, and I think they're the only ones at the moment.
Park.
Oh, Westgate Park. How could I forget wonderful Westgate Park? That's a really interesting area. Thanks, Trevor. Your screen's off, so I didn't know you were there. Yeah, so Westgate Park's a really interesting area under the Westgate Bridge, and they monitor a number of lakes there, so that's quite interesting. Actually, they were just profiled on Gardening Australia a couple of episodes ago, the actual Friends of Westgate Park, not the Waterwatch component, sadly, but the Westgate Park. So that's another interesting area to monitor as well. So the best thing to do is look at that link that I sent and just email the leader of the group. It also has when they mostly meet. So most of them do meet on the weekend. I think Peter Grenfell at Darebin Parklands meets during the week. I think he's the only one that meets during the week. But the rest of them meet on a Saturday or Sunday morning. And just email them and say, "Hi, leader, I'd love to come along and see what you're doing." And you can also tell them that you've started the Waterwatch training, so that they know that you've got a little bit of a background. In regards to the training, just while I'm thinking of it, we normally give you a certificate of participation once you've done the full training, and then we've got a bit of confidence in you that you can go out and join a group and start helping them out. Because we haven't done the fieldwork component and we haven't really worked out a way how to do that online yet, you can't actually get your certificate until you've done the fieldwork component. So after today, when we're allowed to, when restrictions ease enough, because I think we're allowed to have up to 20 participants outdoors at the moment, so we're pretty close. We normally have up to 25 at our fieldwork days. I will actually send out to all of you, everyone that's registered, even people that didn't turn up today but they registered, will get an email with a date of when we do the fieldwork component. Someone's asked about Steel Creek. Yes, I think there are monitoring groups there, but the best people to talk to there are Melbourne Water. So I'll add that into the chat. There's I think it's Waterwatch at melbournewater.com.au, I think, isn't it, Trevor? That you can just email the Melbourne Water crew, and they'll tell you where the groups are. They don't seem to have a web page like we do at Mary Creek Management Committee of where all their groups are situated, so I'm not sure. Are there any groups in dire need of members? Yes, Stephen. Westgate Group are really keen. So the Westgate Park under the Westgate Bridge is definitely keen. So contact Ying for that group. Also the Reservoir Frogs. So that's the group that monitor on the Edgars Creek, just north of Edwards Lake. They've been doing a bit of a recruitment drive, so they're really keen. They're probably the 2 main ones.
One of the groups, the Edgars Creek group, at Ronald Street, which is downstream of Edwards Lake. I don't usually get many people there, so I could always have some extra people there. I think I don't get many people because it's 9 o'clock on a Sunday morning. But,
yeah, I wouldn't be there.
If anyone wants to come along to that, they're welcome.
Excellent. But pretty much all of the groups there, Stephen, on that link, unless they say, there's a couple of groups that say, please, we don't want any monitors. I think there's one group that says that. The rest are all pretty keen because people will, it's volunteer. People might stay for a year and then they move on, do something else. So there's no groups that have like 20 members and there's nothing for you to do. They're all pretty much pretty keen. Okay. Oh, count you in for Coburg North, Kate. There you go. You've already got someone, Trevor.
Yep.
Awesome. So I think that's all the questions for the moment. Thank you, everybody, for those. And we can start the presentation with Trevor with the water bugs. So I'll share my screen, Trevor.
All these phys-chem factors that we've been talking about this morning are important because they relate to the animals living in and around the creek. So what we're really interested in is making the creek environment healthy for anything that lives there. Now, that's anything from platypus to fish to frogs to the birds living around the creek. They're all affected by that. One of the things we can measure is the macroinvertebrates, which is the small insects and crustaceans and that live in the creek. They're also affected by all these factors. And they're interesting because there's something that we can actually collect and identify and use as a measure of the water quality. So although we do platypus survey and we do frog surveys and that, we can also do the work on the macroinvertebrates. And this was all started back in I think it was the late 80s or 90s. There was a guy called Bruce Cheeseman who went all around and looked at what macroinvertebrates we found in various waterways and decided that you could use that as a measure of the water quality. Because there's some of them that only live in really pristine, clean water environments, and there's some that are very tolerant of just about any form of pollution. And so what he did was he went around and started doing all this measurement that's been followed up, and they were able to create a form of collecting these macroinvertebrates and finding out what was in there and giving a score to that, which gives you a score of the quality of the water in the creek. So just want to go to the next slide.
Yep.
All right. So this score, they ended up coming up with what they call the signal score, which is the stream index grade number average level. I think a lot of that was just...
recording in progress.
That's to come up with a nice name. Now, the way it works is that every family of animals is given a signal score from 1 to 2, which means that they're very tolerant of any sort of form of pollution, going up to as far as 10, which means they're only found in the most pristine environments. And there's one bug called a blef, which is a larva of a type of fly, which is only found in waterfalls in alpine areas, and that has a score of 10. So you're not very likely to find that down around Melbourne, but there's a whole range that you will find around Melbourne. Now, because most of these are insects, as you know, insects don't tend to hang around in winter. So when we're doing a sampling to get an accurate signal score, we actually do it twice a year. Once in sort of late spring, which gives them time for the water to warm up and then to breed up and start growing. And once again, usually late autumn. So they're the two times where you can do a macroinvertebrate sample and get an accurate signal score for your particular strain. Any of the groups that I work with, I encourage them to collect macroinvertebrates as often as possible, simply because if you just do it twice a year, in between your sampling sessions, you're going to forget what they all look like and how to identify them. So the more often you do it, better. But to get your accurate score, it's just those two times during late spring and sometime in autumn.
Can I just ask a question about the signal? score. Does that, you mentioned that was sort of, if you like, indicate sensitivity to pollution. Does it also include stream sort of physical things like stormwater pulsing and that sort of thing, which sort of pick up animals and drive them out of the stream?
The score that you end up getting for the stream, once you've done all your sampling and identification, will pick up those influences. What the signal score is supposed to do is give you an indication of the human influence on that waterway. So something like pulsing from stormwater is going to obviously affect your score that you get on that particular day. And as you say, they all get washed away in an urban waterway after a heavy rain event. So if you go and do a macroinvertebrate sample soon after a heavy rain event, you're going to get a really low score simply because most of them have been washed away and they haven't had time to replenish. Okay.
Thanks very much, Trevor.
Great question, and I think it does also remind me that it's really important to, when you're doing water bug sampling and you're looking at your results and to interpret them, having a look at your water quality data as well, and having that knowledge of your stream will really help interpret what's going on as well.
Yeah, and that brings up the point that if you're looking to get an indication of what your stream quality is like under normal conditions, you don't go out and sample 2 or 3 days after heavy rain because you're not going to find very much.
Plus, it's not safe.
And it's not, it's not safe.
In an urban environment anyway, yeah.
Well, yes, it's not safe. So you're not going to go in and collect a sample because it does actually usually involve getting into the creek with a pair of waders on. So if the creek's very high, no, you don't go and do it then. But also it's not giving you a good indication of the normal levels of these creatures in the stream anyway. Anyway, go on to the next screen. So the other thing that's probably worth mentioning at this stage is if you've decided to do a macroinvertebrate survey on a particular day, you want to make sure that you're going to be able to do it comfortably. It's going to maybe take you a couple of hours. By the time you've collected them, sorted them into your various trays and that, and actually identified them, you could end up spending a couple of hours. If there's a lot there, you might even get up to 3 hours spent doing that. Now, you've got to think you're going to be sitting out in the environment somewhere for that amount of time. If it's 40 degrees, you're not going to be very comfortable doing it. Or if it's raining at the time, you're not going to be very comfortable doing it. So you've got to think about, if you're going out on a particular day, am I going to be happy sitting there for a couple of hours doing this? And if you don't think you're going to be able to do it, don't go out and do it because you won't do a proper survey anyway. That's a really important thing, to make sure that you are actually going to be comfortable for the time you're doing it. Now, the actual sampling, what we do is we go down to a stream, you pick a length of or a section of your creek or your river that you're going to sample from. And that's up to each individual group. You look at your stream and you want to pick a section. But what you're looking for is a section of the stream that gives you as many different habitats that are indicative of that stream as you can find. And within a stream, there are a huge number of different habitats. There's the vegetation that grows on the edge of the stream and emerges out. There's vegetation that's growing in the water, and there's different bugs that will live in those. There's the substrate, the bottom of the stream, which has a whole lot of different habitats. There's the substrate at the bottom of the pools, which is fairly calm area. But there's also what we talked about earlier, the riffle areas, those rocky areas the water's flowing over and it's very turbulent. There's a whole range of bugs that specialise in living in those riffle areas. In riffle areas, you've got a problem. It's always turbulent in a riffle area, so you're likely to get washed away. So these bugs have got to find ways of staying in place, which usually means attaching themselves to the rocks. But at the same time, if you're living in a riffle area, you don't have to worry about the oxygen levels in the water because that bubbling and the turbulence creates lots of oxygen. Whereas if you live in some of the pools in a stream, it's much calmer most of the time, unless there's a flood event coming through. You usually not have to worry about getting washed away. But particularly in summer, those pools can actually become low oxygen levels because there's not the turbulence going on. So those bugs have to worry about how do I survive in a low oxygen level. So you get two different communities living in there. The surface of the water is another important habitat. There is a whole range of bugs that specialise in living on the surface of the water, and they often feed on things like butterflies or mayfly, adult mayflies or caddisfly larvae that fall onto the surface. And if you've ever seen a butterfly land on the surface of the water, if its wings touch the water, it gets stuck by the surface tension and can't move. So there's a whole range of animals that feed on things that get stuck on the water surface, as well as a lot of fish and birds and that feeding on them as well. There's the actual water column. There's some bugs that live within the water column, not necessarily amongst the vegetation, but amongst the water column. Another thing is leaf packs, bundles of dead leaves that fall into the water and get caught in little eddies and that in the bottom of a stream. There's a whole range of macroinvertebrates and bugs that specialise in helping the breakdown of those leaf packs, and so they will live amongst there. Also logs and other debris that falls into the water. There's a whole range of things that live on those. So you've got to look and try and find all the different habitats within your section of stream and sample from all of them. And what we talk about is we sample 10 metres of stream, so a 10 metre area of the stream for 10 minutes. But it doesn't have to be 10 consecutive metres. It can be a metre in one area where there's a leaf pack, and it can be a couple of metres in another area where there's a whole lot of vegetation. It can be, a couple of metres of a riffle if you've got a riffle in your area. So the first thing you do when you go down to your stream for the first time, after you've identified the section of stream you're going to sample from, you do what we call a habitat survey. And you actually look at the stream and you allocate percentages of how many different habitats. If it's a quarter of it is made up of riffles, then you say it's 25% riffle, it might be 30% edge vegetation, there might be 5% of leaf pack, and you go back and you get a measure of all the different habitats and what percentage of them are in your section of stream. And that then tells you how long you spend in each of those habitats. If it's 30% of it is riffle, you then spend 3 minutes sampling in the riffle. If 50% of it is edge vegetation, you spend 5 minutes collecting from the edge vegetation. So that's the idea of the habitat, basically just to make you look at the stream and decide what habitats are there, find all the different habitats, and then allocate your time to doing that. All right, next. All right. Once you've collected your sample, you've then got to go and sort them out. So you've gone out with a net. You usually collect it all with a net, and you've scooped through your various areas, and you've got a net full of, usually it's lots of sticks and plants and mud and rocks and a whole lot of animals living in there. You then tip it into your tray, which you can see in the photo, and you've got to then sort them out. And if we, during our field day, we'll actually do a bit of this, and I'll give you a bit of practice, but you spend about half an hour sorting through your tray, and you need to spend a fair bit of time because a lot of them will hide amongst all the debris and that you've picked up, and they'll take a fair bit of time before they finally crawl out. And sometimes you'll sit there and look at a sample for 20 minutes and you'll think, oh, I found everything. The next minute, a dragonfly that's 2 inches long will crawl out. And you'll think, how did I miss that? But they've been hiding, and a lot of them are really good at hiding. So you spend at least half an hour sorting through your tray, collecting all the different ones you can find, and then putting them into your little ice cube tray with a bit of water. And you put the same ones in the same cells of your ice cube tray. And once you're happy that you've collected enough, which is usually after about 30 minutes of sorting, if you haven't found anything new in the last few minutes, you can then stop. But if you find something new after 25 minutes, you add, what we say is you then go and continue sorting for another 10 minutes until you've gone for 10 minutes without finding anything new, and then you can usually stop. And that's when you then start doing the identification. So next screen. All right. We then want to start doing the identification of them. Now, to get a good signal score, you normally have to identify things down to family level, which in the past has been not an easy process. It usually requires taking the bugs out, putting them into alcohol to kill them, taking them back to a lab, looking at them under a compound microscope and counting how many legs are on the legs, or how many little prongs are on their back or something like that. A very technical process, which you can't do without a lot of training and without the right sort of equipment. So there's two alternatives. One, it's usually fairly easy to identify the bugs down to order level, and you can usually do that with a bit of practice. You can do that by just looking at them. And you can get a rough indication of the water quality by measuring down to order level, but it's a fairly rough measure. It's better to be able to get down to family level. And what Waterwatch did is they employed John Gooderin to come up with what we call the ALT guide, the Agreed Level Taxonomy Guide. And that's a way of being able to look at a sample of live bugs in a tray and using things like the way they move, their general appearance, the habitat you've collected them from, all that, to identify down to family level with live bugs. Or if there's a whole group of families that have a similar signal score, you end up with a group of families. But it's a measure that generally down to family level gives you a good signal score, but doesn't mean that you have to have a microscope and a lab set up to do it. So we've now got an ALT guide, and it takes a little bit of practice. It's a dichotomous key. So if any of you use dichotomous keys, it's basically just a series of questions, and you work your way down till you get to a final answer.
Can I just add in something there, Trevor?
Sorry?
Can I just add in something? Yeah, sorry to interrupt. But I just want to mention to people too that we do have a lot of training to get you to this level so you feel confident, and we don't put any of the data, the water bug data, onto either the water bug Blitz app or onto the Waterwatch portal. We collect them in both areas, the information, unless we're confident of your results. So you're obviously heavily supervised when you start. There's lots of different training that you'll do. So when we do the fieldwork component, that'll be a real introduction. But there's Melbourne Water and Waterwatch Victoria run regular, and the Water Bug Blitz now too, which is a national program. So all this data is collected nationally now, which is very exciting. They all run a lot of training. Most of it's free. And look, it's really exciting if you can get out of Melbourne and go into these much more less urban impacted environments, because you get some amazing diversity. And I mean, Trevor's going to talk about the different families of species and the orders of species, but a lot of the things we just don't get to see on the Mary Creek. So if you just continue to do water bug identification, even if you were doing it for years like Trevor has, and you just did the Mary Creek, you're not going to have a really good breadth of experience, because you're just not going to see some of the more top order water bugs that live in less polluted streams. You're just not going to have that breadth of knowledge. So it's a really interesting science. And I think the other thing is what John Gooderin set up from the Water Bug Blitz is a system of training. So it's a bit like the karate belt system, blue belt, black belt, etc. So you start off on, is it yellow belt?
Yellow,
or orange?
Yellow belt.
Yeah, yellow belt, which means someone supervised you in collecting the sample properly, and you can actually sort it out into the trays, and you can do a little bit of identification. And it goes all the way up to black belt. where you basically can go and you can train others, and you've got a really good knowledge of all the different diversity of species from families. And of course, by the way, the alt guide, some of the species we can get down to genus and sometimes even to species if there's not a lot in that group and they're easier to identify. So it's a really interesting area. Sorry, Trevor, I'll let you keep going.
All right. So thanks for that. So we've got the alt guide, which there's a link there that can be freely downloaded from the Waterbug Blitz site. The only thing is that it's how many pages is it? About 50 odd pages. So if you print it out, it's a fairly big printing job. 100 pages. So if you print it out in colour, it's a fairly big thing. I prefer to use the paper version myself because that's what I'm used to. But there's also the Waterbug Blitz app, which is basically the same guide, but it's in an app form. It's actually really quite useful because you can go through, when you use it, you actually put in the first thing you put in your data is where you are, the name of the stream. You take a photo of it. It also gives them a GPS location of where you're sampling. You do your habitat survey on the app as well, so that helps you identify the various habitats. And then you can sort of electronically add all the things. That comes up with a little picture of an ice cube tray, and there's a key there and helps you identify all the bugs. So the Waterbug Blitz app is just a really easy way of entering all this data. So you've got all those. The other thing I'd mention is John Gooderham and Eddie Serlin years ago wrote the Waterbug book, which is just a good general reference for if you're interested in the water bugs and something about their ecology and lifestyles. So that's another useful resource. But there's lots and lots of helpful resources you can get on. There's Waterbug Face on Facebook, and if you put in a picture of something on Facebook, someone will get on and tell you what it is. So there's lots and lots of help for people who are just starting out.
Especially the Waterbug Blitz, Trevor, isn't it? And you can see on the PowerPoint presentation we've got that. That's the app there, but the Waterbug Blitz, if you just Google Waterbug Blitz, that website's got heaps of resources, and there's even John Gooderham doing lots of YouTube clips now as well with collecting the sample and identifying the, sorting it out, all that kind of thing. I mean, obviously it's not as good as being out there in the field, but for the moment it gives you a bit of an insight, gives you a bit of an idea of what to expect when we all go out into the field. So, feel free to have a look at those as well.
Now, earlier on, you were saying about for people collecting in urban areas, there's not nearly as much diversity in the water bugs, which is very true. The only advantage of that is that in your local waterway, you can usually find with a bit of practice, you can quickly identify all the bugs in your area. So yes, you're not seeing all this amazing range of bugs that you'll find up in a mountain stream or something. But what you can do is quickly get to the stage where you can be comfortable with identifying the water bugs in your area. And that doesn't take as long as learning to do everything takes. I love to go out in the mountain areas and sample from there, and I just find a huge range of bugs. But I might spend 3 or 4 hours at a stream just trying to identify them all. So there's pros and cons, but if you're in the urban areas, yes, you can still do this and you can learn fairly quickly what your bugs are. Move on to the next one.
Yeah, really good point, Trevor.
All right, we'll start. What I'm going to concentrate here is the sort of things you're going to find in urban areas. I won't go into all the range of bugs in other areas, but we'll start off with some of the ones with low signal scores that you'll find, and we'll sort of work up. Now, some of the lower ones you will almost always find just about anywhere are things like up in the top right-hand corner, there's an aquatic worm. It's basically just the same as an earthworm, except they tend to be only about 1 millimetre or 2 in diameter, but they live in the bottom of streams. They're usually crawling around the mud in the bottom of streams. Often we'll find them. Bottom left-hand corner is a little bug we call a bloodworm. They're the larvae of chironomids. Chironomids are non-biting midges. So any of you on a summer's day have walked down beside a stream or a creek and gone through a cloud of tiny little insects that all get up your nose and in your eyes and everything, that's the adults of these chironomids. So they're non-biting midges, so they don't do you any harm, but they're a really common creature living in the bottom and the sediments and bottom of just about any stream. The red ones are actually red because they've got hemoglobin in their system, the same as we've got. That allows them to live in muds that have very low oxygen levels, which is good because there's lots of food down in the mud for them to crawl around and eat. But also you've got to think of, if you're a bright red, wriggly, wormy thing, there'd be a lot of things would like to eat you. And living down in the mud where there's not much oxygen, again, gives you a bit of extra protection from all these other things that want to eat you. The bottom right-hand corner bug is called a blackfly larvae. Now, this is one of our specialist riffle area bugs. I've put in the chat a link to a video that shows you these guys moving, but what they do, some people say it's like a leech movement, but I think more it's like a caterpillar, where they put their head down and then they bring their back up. So the head is the little dark bit at the bottom, and the bulbous bit at the top is actually their tail. And they put their head down, and what they do is they don't have suckers like a leech does. They actually weave a little silken mat on the surface, and then they bring their tail up, and the tail's got a whole lot of little hooks on it, which they grab onto the little silken mat, and then they go forward a bit, put a few more silk threads down and bring their tail low. So it's very much like that caterpillar movement that you see with a caterpillar walking around on a tree. These guys specialise in living in riffle areas, and what they'll do is they'll weave a little silken mat onto a rock, use their hooks to hook onto that silken mat, and then put their head up into the stream of the water, and their antennae have been modified to form little fans, and they collect bits of organic matter and that flows past and put it into their mouths. So most of the time they're just sitting there on a rock, waving their heads around in the water and eating anything that comes past. So, but in your sample, once you've seen them and seen the way they have that little caterpillar movement, you'll identify them very easily. But they're only about 5 millimetres long, so they're quite small. All right, next one. A couple more things that we tend to find that are fairly tolerant of any sort of situation. Leeches, there are small leeches that live in most of our streams. Most of them don't suck our blood. They're actually usually attacking fish or other macroinvertebrates. I know there was one day I was sampling up the wetland near Edgar's Creek. There was all these really big diving beetle larvae. They're about 2 or 3 inches long, huge larvae. And some of these had about 6 or 8 leeches attached to them, which, so they were feeding off these other macroinvertebrates. So yes, there are lots of leeches. Again, they're fairly tolerant of any sort of situation. The one in the middle is called a flatworm, Turbularia. Often find them, they again crawl around on the surface of rocks or vegetation and that, scraping off the bacterial layers there. But you'll see them in the bottom of your tray. They're just sort of like a tiny little flat thing about 5 millimetres long, sliming along the bottom of the tray. And the one on the right-hand side is a pond snail. These are actually an imported, basically they're a pest, but they're now all through our streams. They're called the European pond snail. They're tolerant of just about any sort of conditions. And the way you can tell it's one of these particular snails is that you can see the motley pattern on the shell. That's a distinctive giveaway that it's one of these pond snails. There are lots and lots of other snails that we find in our streams, some of which only tolerate very clean waterways, some of which we tend to find occasionally in our urban waterways. So there's a few more than just the pond snail, but this European pond snail is by far the most common one in our urban areas. Next. Right, next we get starting to get up into other creatures that are a little bit less tolerant of pollution. Now, most of the bugs we're going to find are insects. So they've got 6 legs, they've usually got a couple of pairs of wings and that. Now, there's two groups that we will commonly find in waterways. On the left-hand side, you've got true beetles, or the order Coleoptera. You can tell beetles from true bugs is beetles have a distinct line going down the middle of the back. You can see it on the one on the left-hand side. Beetles, or a typical insect has 2 pairs of wings. What the beetles have done is one pair of wings they've actually turned into wing covers. And what you're seeing there is the front pair of wings that are modified to form a cover which protects the rear pair of wings. So they only fly with 1 pair of wings and they use the other pair as protection. And when they fly, that front pair opens out and that line going to the middle spreads apart, and then the other wings come out and they can fly. The other order on the right-hand side is the Hemiptera, or the true bugs, and you can see that they don't have that, although this one's got a bit of a line going down, the line doesn't go all the way through. And what these guys have got is 2 pairs of proper wings and they're folding over. So it's an asymmetrical appearance on the back. So that's how you can tell the difference between beetles and bugs. Both of them have adult stages that live in water. There's also, with both of them, there's lots of members of these orders that don't live in water, but both of them have got adult stages that live in water. If you go to the next slide, if we go into the beetles, beetles will also a lot of the time have a larval stage that lives in the water as well. So you can see that here. On the right-hand side, we've got a diving beetle, which they're fairly distinctive. If you're looking at it and you see a beetle and it's only swimming with basically its back pair of legs, which you can see clearly in that picture, they're the diving beetles. They have a larval stage that lives in the water, which looks very different. And this is the larval stage of the same thing. The larval stages are predatory animals. You can just see the front of it. He's got a pair of pincers there. So he goes around biting just about anything he can find.
That's a nasty set of pinchers there, isn't it? Ooh.
Nasty set of pinchers. If you're only a little bug, you don't want to run into him. So beetles will have both an adult and a larval stage in the water. If you go to the next slide, the true bugs tend to only have, they don't have 2 distinct stages. The larval stages are basically just small versions of the adult. And we've got a range of bugs here. Top left-hand corner is called a water treader. And I was talking about bugs that live on the surface of the water. This is one of them. They're only tiny. They're only a couple of millimeters long at the most. And you will often see just this little black thing running around on the surface of the water. You collect them. They're often hard to keep in your tray, so you need to remember that you've seen them, because you put them in your little ice cube tray and they just crawl out and fly away. So you've got to remember those. So they're very common. And they're, again, one of these things that are waiting for things to fall onto the surface of the water. So anything like a mayfly adult that's fallen on the surface, gets trapped on the surface of the water, all these little water treaders will rush out there and basically attack it. And they've got little probes and they suck the juices out of the body. The top right-hand side is the needle bug. These are fascinating creatures. We sometimes find them. They're quite big. They can get up to probably 10 centimetres long.
Do they prefer slower waters, Trevor?
Yeah.
I seem to find them in wetlands more than the creek.
They tend to be in wetlands, although you'll find them in sort of the edges of stream amongst the vegetation. And they're actually a predator. The little arms on the right-hand side of that picture are the front legs, and they act like the legs on a praying mantis. So they've got the ends of the legs hook around, and they can grab things and pull them in like a praying mantis does. The tube on the left-hand side there is actually a snorkel tube, and that allows them to sit under the water and put this above the surface of the water and breathe, because these are actually air-breathing animals. They don't have gills, so they have to be able to breathe under the water. They can take in some oxygen through their outer shell, but that's not enough to keep them going for very long. So they have these long tubes to sit that above the water and use that for breathing fresh air while they stay under the water. And they're just a stealth predator. They'll sit there amongst the vegetation, wait for something to come past, grab it and eat it.
And it really shows, I guess, they can, live in a bit more of a variety of habitats with low oxygen because of that breathing apparatus they've got.
Yeah, and that's the thing with a lot of beetles and bugs is most of them are actually air breathing, and they've come up with different ways of doing that, and that allows them to live in some of the less pristine environments. The bottom left-hand corner is a water boatman. Again, very, very common. You'll often find that these are one of the first colonisers of any water body. So if you've got something like a swimming pool that hasn't been chlorinated for a while, the water boatmen are one of the first ones that will come in and inhabit that. The way they do that is they're again air breathing, but the way they get around it underwater is they attach a bubble of air to their underside. So they come up the surface, they attach a bubble of air. I think they've got a whole lot of little hairs under their body, which allows the air to stick to their body. And they use that like an aqualung when they're swimming around underneath. But when they decide they don't like where they are anymore, they can crawl out of the water, dry off and fly away and go and find a new place to live, which is why they're quite good at inhabiting or colonising new water bodies. The bottom right-hand side is a back swimmer. Much the same thing. They're air breathing, so they attach a bubble of air to their body for swimming underwater. The thing that gives them away from the water boatmen is they tend to, first of all, they're called back swimmers because they swim upside down. So if you see something swimming upside down, it's probably a back swimmer. Also, the two legs there, you can see they have long, thin legs coming out the front, which they use as oars. The water boatmen on the left-hand side there, the two pairs of legs are modified, but they've got more paddle-like, so they're a little bit flattened, whereas these are just long and thin. The back swimmers, why do they swim upside down? Well, they're, again, one of these creatures that's hunting for things on the surface. So they're continually looking up, looking for something that's stuck on the surface of the water, and they will then swim up and attack it from underneath. So the surface of the water is a really bad place to live if you're a little bug. All right, next. All right. A few more of the bugs that are sort of in the mid-range. Top right-hand corner, there's quite a number of little freshwater mussels we can find. These ones get up to about an inch in diameter. But there's another little shell that we often find. It's called a pea shell, and they're only probably 3 or 4 mils across in diameter. So they're very hard to find because they often just look like a little sand grain in the bottom of your tray. But if you look hard enough, you'll you'll start to notice that there's some things that look like sand grains, but then you look close and you go, oh, that's not a sand grain. Oh, and you look a bit harder and, oh, that's a little shell. So these little pooey shells are very, very common. The bigger ones that are shown here are less common to find, but they're still found occasionally in our waterways. Bottom right-hand side is a water mite. Find them fairly commonly. They're very small. They're only 1 millimetre or 2 in diameter, but they're often brightly coloured. So if you see a brightly coloured blue or red spot swimming around in the water, that's probably a water mite. And you look close enough and you'll see the little 8 legs swimming around. So again, a small one that you can, with a bit of looking, you'll usually find. But again, they're more of a giveaway because they're often this bright red or blue colour. And freshwater shrimp, glass shrimp, we often find they're a bit sensitive to pollution, but we still find them in the Merri Creek. So I think most of your waterways in Melbourne will have them. But again, it'll usually be more likely to find them after drier periods where there hasn't been any big influxes of rain to wash them away. All right, next.
Got about 4 or 5 minutes left too, Trevor, just to give you a...
Right, yep. Next, dragonflies, the order Odonata. You've got dragonflies and damselflies, 2 common inhabitants. Damselflies tend to be a bit more common. Difference between them in the adult stage, bottom picture on the left is a true dragonfly. When they tend to be more robust in the adult stage, and when they land, they tend to hold their wings out. Damselflies are usually more delicate, and when they land, they tend to fold their wings beside their body. So although they look very similar in the adult stage, you've got those 2 distinguishing features. In the nymph stage, which you find in the water, they look very different. The damselfly on the bottom right-hand side, they're usually much smaller than the adult, than the dragonflies, and they have these 3 tails sticking out the back. Those 3 tails are actually their gills, so that's what they breathe with. So you've got them. The true dragonfly nymphs on the left-hand side there, their gills are actually internal in the abdomen. So they can suck water in and out of their abdomen to aerate the gills, also allows them to do this little jet propulsion through the water. But they also tend to be, like the one in the diagram there, that's an ashnid dragonfly nymph. They can get up to 30, 40 millimetres long. So they can get quite large before they crawl out of the water and turn into a dragonfly. Both of them are predators. They've got these amazingly modified mouth parts where the lower lip is hinged and can shoot out. And I'll put a link onto the chat later on showing you a little video of one of these guys hunting.
That's really cool, that one, isn't it? Because they've slowed it down, the actual, how the mouth comes out and grabs things. So it's pretty amazing.
Again, if you're a little bug living with dragonflies, they're just ferocious hunters. All right, next. All right. Next, we get into these 3 orders: the Ephemeroptera, the mayflies, the Plecoptera, which are stoneflies, and Trichoptera, which are caddisflies. Now, this is where you start to get into the areas of getting really high signal scores if you're starting to find lots and lots of these animals. In an urban area, the stoneflies, the one in the middle, you're just not going to find them around Melbourne. They are really only in less affected areas. But the nymph stage of these, the difference between mayflies, which are on the left, and stoneflies in the middle, you can see, although the one stonefly you can't see, the mayfly you can see has got 3 spiny tails. So that's mayflies. Stoneflies have 2 spiny tails. So if you see a creature with just the 2 spiny tails, That's a stonefly. If it's got 3, it's a mayfly. The only thing is you have to be a little bit careful when you're collecting your sample is the tails tend to break off. So sometimes you find mayflies that have only got 1 tail and 2 little stumps sticking out the end. You just got to be a little bit careful of that. So stoneflies you're not going to find in Melbourne. You really have to go up the other areas. Mayflies, occasionally we find 1 or 2 families of mayflies in Melbourne. The one that's in this diagram is called a baited mayfly, and we occasionally find them in the Merri Creek. They're really distinctive. They only grow up to about 1 centimetre at the most in length. And when they're in the tray, you'll see them, they swim very rapidly in these short little bursts. So if you see a little thing with 3 tails that swims in a really short burst, that will be a baited mayfly. But very few. There's lots and lots of different families of mayflies. There's only 1 or 2 that you'll find in urban areas. On the right-hand side, we've got the caddisfly or the Trichoptera. Now, this is one of the main orders of aquatic insects. There's thousands and thousands of different species of Trichoptera around the world. I think something like 11,000 species that I've identified. And the vast majority of them are only found in fairly pristine environments, but there are a few that we tend to find down in the urban areas. So if you go on to the next screen.
Yeah, got about a minute left too, Trevor.
All right, this is some of the caddisfly that we find in urban areas. Now, caddisfly, there's 2 basic groups, although they're all caddisfly. There's what we call free living, which just live without a case, and there's cased caddis. Cased caddis make a case to live in, but the actual bug looks much like the free living ones, which don't live in the case. Now, the free living ones I've shown you there, the one at the top there on the left-hand side is called a net spinning caddis, which is family Hydrocycid. We often, well, in fact, we nearly always find them down the Merri Creek and the riffle areas. They're distinguished by having gills just under the bottom of the abdomen there. You see the little fluffy bit on the abdomen. That's its gills. They live in riffles and weave a silken net which collects organic matter, which they can then eat. They're a real giveaway in your tray because being a riffle specialist, they like to have high oxygen levels. As soon as you put them into a still tray of water, they start to have trouble breathing, so they thrash around. So if you see a little thing that looks like that with sort of 6 legs, long body, and these sort of little hooky tails on the end, and it's thrashing around wildly, it's almost always going to be a net spinning caddis, particularly if you're collected from a riffle area. The one at the bottom left-hand side is an ecnomid caddis. They're not a riffle specialist, and they'll just tend to be crawling around on the bottom of the tray. They're often given away on their head case. The body is white, but the head case is a bit yellowy and often with a brown mottled pattern on it. So if you see something which got a brown head with a mottly yellow pattern on it, that's probably the ecnomid. Right-hand side, we've got 2 of the case caddis, which we commonly find. At the bottom is a genus, actually. Well, both of these are from the family Leptoceridae, but this is one of the cases where we get down to genus level. The bottom one is Notalina, which we call them headbangers because when you see them in the tray, they put their head out of the case and they thrash their head up and down to swim, and it looks like they're headbanging at a rock festival. They also make a case by getting a bit of plant material and weaving it into a spirally cone, which you can see in the picture. The one above is, again, it's Leptoceridae, the genus Triplectids, and they're just called stick caddis because they just get a piece of stick and live in it. They don't actually weave it from a piece of plant material like the one at the bottom. So, again, that's one of the ones where we can get down to genus level. So, given that we're running short of time, I'll shut up there, unless anyone's got any questions.
There's quite a number of resources. If you go to the Waterwatch Victoria website, there's actually quite a number of resources. The Here and Now Waterbug book. Melbourne Water has a small field guide as well. The Alt Field Guide, the alternative level taxonomy that Trevor's mentioned. So there's quite a lot of resources out there. I've also got some worksheets that I work with schools and things that have some really good drawings as well that I'm able to share as well. So there's quite a lot of information out there, and if there's anything you would like, you're welcome to request it from me. And thanks to Trevor for that. I'm sure you'll agree it's very engaging talk, and you can just see how passionate Trevor is about his bugs and his knowledge. I always learn something new. It's so fascinating, and I can't wait to get out and look at the trays again. So we will definitely be doing it in the fieldwork component, but Trevor and I are also planning so this is a regular site that we have. We've got a number of regular sites on the Merri Creek that we go back each year. We try and go back at least once a year, more if we can, where we collect data for council. So it's going to be at Merri Creek in Coburg near Deschene Reserve, so that will be in spring. So if you're interested in knowing about that, the best thing to do is subscribe. So I'll also add this to the email. We have a subscription list on our Merri Creek Management Committee website, and you put your name and your email in and then tick the box that says events like Waterwatch training. So that's anything to do with Waterwatch, like whether it's collecting litter, whether it's water bugs, whether it's water testing, whether it's Waterwatch training, the frog census events that we run, all those types of things. There's even a tick box for frogs if you're interested as well, because we do a lot of frog stuff. So just look out for that email, that subscription, if you want to be kept informed of any water bugs training we do. Melbourne Water also run water bug training sessions. So it's a good idea to just go to the Melbourne Water. If you just Google Melbourne Water water bugs, it'll take you to their citizen science Waterwatch area, and they've got an email list as well. That's a really good one because our sites, we just go mainly the Merri Creek catchment because that's sort of where we're funded to go, but the Melbourne Water Group can go Greater Melbourne. So, you know, there've been trips out to Warburton, I think Ledyard Gorge, what was that lovely site near Macedon? Is that right, Trevor? We went Garden Hut Creek, I think it's called.
Oh yeah. No, that was actually it was near Macedon.
Yeah, so
it's their private property though.
Yeah, but that's, you know, because Melbourne Water's organised and they've got good relationships with this people that own the private property. We've got to see some amazing bugs there. So if you're on that email list, you'll hear about different events like that you can go along to. And look, Trevor and I have certainly taken volunteers along. We carpool where we can, and we go to these sites for the day, for a day trip. But there's also opportunities to go for the weekend. You know, I know of some Waterwatch monitors that have really got into it and want to go up in the black belt levels. So to do that, to get better, as Trevor mentioned before, you've got to keep doing the training and go out as much as possible. So there's been people that have made a weekend of it. They've gone up different parts of Victoria, done the water bug sampling and made a weekend of it.
I often, when I can, I go out to the country and I like to go camping, and sometimes I like to camp beside rivers, and if I'm going camping beside rivers, I like to collect water bugs. And I strongly encourage other people to do it. But if you are going to go and do that, you need to be aware of the cleanliness of your equipment. It's really important. When you're doing sampling down around the urban areas, the waterways you're sampling out of aren't pristine. There's a lot of pollution, a lot of diseases in the water. If you're going to go and use the same net into a pristine alpine area, you need to make sure that you've cleaned it. And that's cleaning it usually means putting it in hot soapy water, maybe washing it down with metho, and also leaving it out into the sun. Drying it out in the sun, the UV radiation kills most of the things as well. But you do want to make sure that you're not taking contaminants from an urban area out to pristine environments, because there's also things like the chytrid fungus, which seriously affects frog species. Some urban streams don't suffer from that at the moment, and if you go and take a dirty net into an urban area, you might be spreading something like that, which is going to affect frog populations as well. So just be careful of that if you're going to go out in the more pristine environments.
On behalf of Trevor and myself, I just want to thank you all for being here, and I hope you've really enjoyed it. I can see in the chat, thank you for those that have put in the chat that they have enjoyed. Thank you so much, and do fill out the feedback form, because we do rely on funding from councils and Melbourne Water to keep this going. So any evidence that we can collect that people are finding it valuable would be really, really helpful for us to keep these going. And have a lovely day, everyone. I'll be in touch, obviously, with when we can do the fieldwork component, and I will send a link to the recording when we've edited it and it's available. Thanks, everyone.
Thank you.